Hello, my name is Ismi Hanuddin. I'm currently a second year undergraduate at the University of Illinois at Chicago. I am also a research assistant within the Division of Endocrinology, hoping to further my career in medicine.
I'll be presenting how glucose reversibly controls protein stability of HKDC1 in hepatocellular carcinoma. And to begin with, globally, we are seeing a decline in the number of deaths due to cancer malignancies. However, the incidence and mortality rates of liver cancer continue to rise, with hepatocellular carcinoma being the sixth leading cause of cancer deaths worldwide, claiming over 12,000 lives annually. In hepatocellular carcinoma, we see an aberrant glucose metabolism, and the novel hexokinase HKDC1 is significantly upregulated in HCC. In previous literature, we have shown that HKDC1 is involved in glucose metabolism, and we have data that HKDC1 is essential for the progression of hepatocellular carcinoma. How HKDC1 expression is regulated in HCC is still unknown, however. And then as background, glucose is a nutrient and a signaling molecule within cancer. Thus, we hypothesize that glucose regulates HKDC1 expression in hepatocellular carcinoma. Now, in this first figure here, we see HepG2, which is a common HCC cell. We vary different levels of glucose concentrations here, ranging from 1 millimolar all the way to 25 millimolar. So this is very low. 5 millimolar is about the normal amount, and then 25 millimolar is a very high concentration and typically present in diabetic patients. When glucose concentrations are low, we see that there's less protein expression of HKDC1. However, there's no significant changes in mRNA expression, nor is there a change in hexokinase 2 expressions, which are also upregulated in hepatocellular carcinoma. So only the HKDC1 is being regulated by the glucose through this model. To further test this observation, we starved the HepG2 cells of glucose from half an hour to 6 hours, and at the end, we performed immunoblotting and qPCR, and our data suggests that there's a significant degradation of HKDC1 in as little as 2 hours, as shown here. To prove that this phenomenon is not only specific to one of the cell lines, the HepG2 cell line, we performed the same experiment to 4 additional cell lines, and in all of them, we observed the same results of HKDC1 degradation as shown here. So the left columns show the presence of glucose, and then the right is the absence of glucose. And in all of them, we see kind of a degradation of the HKDC1. To further test if glucose was added back, can the HKDC1 expression be reversed? So we starved the cells of glucose for 4 hours and then added back glucose concentrations from 2 millimolar to 25 millimolar again, and saw that the degradation of HKDC1 was in fact reversible. Now, due to the fact that this is protein degradation and not related to mRNA, we assessed how HKDC1 is being degraded. And there's 2 main factors we consider within protein degradation, which is whether it's autophagy or proteasomal degradation. So to test this, on the left side, we have different inhibitors of autophagy, and none of them were able to sort of rescue the HKDC1 protein. However, on the right side, we have MG132, which is an inhibitor of the proteasomal degradation, and in lanes 3 and 4, we see that there is a rescue of HKDC1 even with the absence of glucose. Thus, we conclude that HKDC1 is degraded through proteasomal degradation mechanism in the absence of glucose. To further confirm this, we used IP to check if HKDC1 is ubiquitinated in the absence of glucose. So we grew the cells with and without glucose in the presence of MG132, as shown here. And we did this to block the proteasomal degradation and followed up with an immunoblot to conclude that in the absence of glucose, HKDC1 does in fact become ubiquitinated. And then down here, we assess the relationship between HK2 and HKDC1 within the protein stability. So we have facetin, which is a glucose transport inhibitor, and then 3BP, which is a hexokinase 2 inhibitor, IA, which is a GAP-DH inhibitor, and this inhibits the reaction halfway in glycolysis, and then 6AN is a PPP pathway inhibitor. And then this shows that HKDC1 was affected by the 3BP and the facetin, indicating it becomes degraded with the inhibition of HK2 and glucose transport. So if we saw these results for the IA, it would be obvious that this is kind of lower down in the chain of glycolysis, but because it's happening in the inhibitors of facetin and 3BP, we conclude it's a kind of a result of HK2 and just glucose transport. So To confirm, we used RNA interference where we silenced HK2 with SIHK2, and we saw a similar decrease within the HKDC1 and HK2. Thus, we've concluded that HKDC1 is regulated by glucose through hexokinase 2. And further studies are needed to figure out which metabolites of glycolysis are responsible for this regulation and which E3 ligase is responsible for the HKDC1 degradation in the absence of glucose. Thank you.